amplified fragment length polymorphism. Means you can see that it's the polymorphism. The basis of polymorphism is fragment length. Means there is a based on the difference in the length of the fragments produced by this marker system, we can able to identify the individuals. If anybody asks you that what is the genetic basis of AFLP system, it's based on the fragment length. You know that the fragment length polymorphism we observed in RFLP also. RFLP. What is the difference between them and this one is here there is no amplification. In RFLP we're not going for amplification of the fragments. But here we'll do amplification. How the process will go, we'll discuss. AFLP marker is based on selective amplification of the DNA fragments using the polymerase chain reaction. In RFLP, we are not using any PCR, but in AFLP, we are using a PCR. How it will, how exactly the process I will discuss. First, we will discuss what is AFLP. And AFLP marker, they do not require prior knowledge of DNA sequence and making them useful for non-model organisms or organisms with poorly characterized genomes. For example, take citrus. Genome sequence is not good. So that at that time we can't go with SSR marker and SNP markers or any other marker which needs sequence information. What are the marker? RAPD and RFLP and this AFLP and ISSR. I will discuss what is ISSR. These are the marker system. We don't need any sequence. You just randomly design any primer or you just randomly treat restriction enzymes either based on fragment length, either based on presence or absence. We will be able to differentiate the individuals. Means the AFLP no need of any sequence information. This point you should remember. If someone will ask you, AFLP is sequence-based marker, either prior sequence information is needed. You say no. There is no need of sequence information. Then AFLP marker can be used to study genetic diversity, population structure, evolutionary relationship between different groups. Marker, because we already studied what is the use of marker. It also used for any studies. Okay, it can use for mapping, anything. Because it's having combined advantage of both your RAPD plus RFLP. It's like a hybrid. Okay, it's a hybrid of RAPD and RFLP. Then it overcomes the disadvantage of both the marker and it combines the advantage of both the marker. Then AFLP marker can also be used for plant animal breeding. This is a general point. We'll discuss what is the procedure. First you have to isolate the DNA. Then you have to treat this DNA with some restriction enzymes. You know what is restriction enzyme? And there you have to use 2 restriction enzymes. Can anybody guess why we need to use? See, here they used one is 4 base cutter, another was 6 base cutter. What is the use of using? What is the use of using 2 restriction enzymes? What is the advantage? Why they are using 2 restriction enzymes instead of single restriction enzyme?
They will create sticky ends with different base pairs.
Okay, you can some sticky the restriction enzymes will produce sticky ends that can also be used. Why 2 restriction enzymes? 2 different restriction enzymes.
Sir, the later we create some primers, sorry, sir, some sequence to link to the regions, sir.
No. We are not making any primer out of this. I told there is no need of any sequence information.
Sir,
Actually,
we will link adapters.
Okay.
They create 2 different ends.
No. Final advantage. I'm asking why to do all those things. Why
Sir,
need to
we
adapt
know
this?
we know the adapter sequence, sir. We create the sticky ends according to the adapter sequence, sir. For those reasons,
Here,
I
no.
think
See,
this.
here they are not doing anything. What is the goal of doing all those things? There is a one terminal reduced representation. Reduced representation means, it is a DNA. Normal you take rice for 430 MB means 430 million base pairs. The probability having if 80 GC is equally distributed, if it is 6 base cutter, the probability having the 6 to the power of 4 times maybe the probability having the restriction sites. Means we have large number of restriction sites. Means we all know this. Many of the genome sequence have restriction set. See how many millions of fragments we will going to generate. We will generate millions of fragments. When you look into the gel, for example, this is individual A, this is individual B. When you treat with single restriction animal, you will get smear like this. Any interpretation can you draw from this? Why you are using marker? You have to differentiate these two individuals. But from the bands, take this as gel. This is a gel and this is a gel. These are the bands. See, you will not get any information from this type of gels. For example, you take another gel, only 2 bands are there, or only few bands are there. This is A. So you can say, okay, this is 50, like this is 100, this is 50, like this it is going. You can say this is some 70 base pair, this is 100 base pair. Okay, this marker is polymorphic. You can say, okay, this is a marker can able to differentiate the individual A and individual B. But if the gels are look like this, can any interpretation can be drawn out of it? Is there any possibility? Abhishek?
sir.
Yeah, there is no possibility. That's why we need a strategy called reduced representation. Reduced representation means you have to reduce the number of bands here. There is a formula. I forgot actually. I will tell the formula. You have to reduce this representation. If you use restriction, the probability having one restriction site is 4 to the power of 6. But the probability having the site having both the restriction sites, say PST1 and MSC1 and PST1. This type of it is very, very low. Anybody getting the point? The probability of getting the fragment having the restriction site MSC1 and PST1 like this, it's very, very low. Means your representation will go, you are selecting, see, further you are selecting this type of bands only. I will say. See, first, the extracted DNA is digested with 2 different restriction enzymes. One is usually frequent cutter, another one is rare cutter. The step to generate a pooled DNA fragments of varying lengths. Then, after that, restriction fragments are then ligated with adapter molecules and contain specific sequence that allow PCR. Then adapter ligated fragment is subjected for, actually I didn't mention that point. We'll select these fragments, then DNA fragments with PST1 three types of you're getting. Then we'll add an adapter ligation with MSC1 and PST1 adapter. We are joining adapter to this fragment only, so that we are eliminating these two. Means your representation will go very, very less. We'll get very few fragments. Then here also, after ligating with the adapter, one with which joins with the PST1, another one is joined with another restriction enzyme. Then after you should amplify this. How we can amplify this means you can use random nucleotide only, but there is some one or two first with one selection nucleotide. Means for example A, T, G, C were there. This A is the selection nucleotide. This primer should have A in this particular. Means wherever the complementary T will be there, it only amplified. But wherever the here opposite T was not there, it will not going to bind. Means you are selecting some fragment. We are eliminating all the fragment. We are selecting only some fragment by using selection nucleotide. Then after that again you have to go for PCR with 3 selection nucleotide. Means if you use one selection nucleotide, for 1000 fragments, you will reducing 500 fragments. If you use 3 selection fragments, you are reducing to only 100 fragments. Like this, we are reducing the representation and we can able to have only fewer fragments so that we will interpret the results well. Means first you have to treat with 2 different restriction enzymes. Then you have to select the fragments which having both the ends of the different restriction enzyme. You can see that MSC1 and PST1, they are having the restriction site for both the restriction enzyme. Why you are selecting this means the probability having the fragment having both the restriction site is very low when compared to probability of having this restriction site, probability having this restriction site, this probability is very low. Means you are eliminating millions of fragments, you are selecting only some fragments. Then you are ready this is one step of reducing representation. Another step is amplifying with the selection nucleotides. One is with one selection nucleotide, another PCR with 3 selection nucleotides. Then further you are reducing this second step of reduced representation. By using 2 steps, we are reducing the representation. Means it is one of the advantage over the both the marker system like RFLP and RFID, where we will get a lot of smear. Bands will like this. I think those who are worked with bands, they will get to know. It's very difficult to understand. They will get smears. This is called as smear. To avoid getting smear, we are using 2 techniques. One is two different restriction enzyme. One should be rare cutter, one is frequent cutter. I think you know what is rare cutter, one is frequent cutter. Then another thing is using PCR with selection nucleotide. PCR with selection nucleotide. These 2 steps is used to reduce the representation, and this then we will detect the then they'll keep to autoradiograph. Then the polymorphism is detected. Understood what is AFLP marker? And it is dominant in age. Okay. Remember, AFLP marker is a dominant, means it's not able to differentiate dominant homozygote and heterozygote. It's a dominant in nature. Any doubts in AFLP marker system?
Sir.
Yeah.
Sir, understood the second step of that reduction, sir. But as you told, the probability of getting a fragment with both the restriction site is rare or less. How would you select that fragment, sir, in first step of dimensity reduction?
We are adding adapters, na?
Yes, sir.
We know restriction. Okay. We know all sequence of restriction sites, na? MSC1 yes, where it will go fine. PST1 restriction set we know, na? Generally yes, we know, na? Restriction yes, sites sir. And their restriction sequence we know, na? Based yes, on that, we designing adapters. Then those will have that adapter only, and they having some selection nucleotide, one or selection. They will only amplify. It's not like that they will select this band and they will amplify. Okay. See, they are having this band end. They will design adapter. See, that's why AFLP specific, like MSCL specific adapter and PST1 specific adapter. They will go and bind here. They will amplify them only. Okay. We know restriction sites of all the restriction enzymes. Easily we can design adapter for them. Okay. Now, any doubt?
Sir,
Your
here
doubt is clear?
adapter. Hello.
Yeah. Ask us.
Sir, here adapter is used as primer.
Yeah. Adapter. No. Yeah, exactly. First, we'll add adapters. Actually, adapter like MSc1 adapter and PhD1 adapter. Actually, for this blunt end, we'll add one adapter. Then for this one, we'll sequence some sequences. Okay. For this, we design primer. Okay. First, we will add some sequence to it. Like actually, it's an extra sequence because here some sequence is missed. Then we will design a primer for this one. Then we will amplify. Okay.
Okay, sir, and that primer
Sir,
is that
But
adapter.
adapter is as complement. Adapter is used as a complementary sequence for primers. Okay.
Yes, sir.
But adapter blunt end sequence is there. It will join there only. Now you understood.
sir,
When you add
why?
MSc1 specific KFLP adapter, it will go join here, and this adapter will go join here. For this, we are knowing already the complete sequence of this one. Adapter sequence also we know. Then that one, from that one, we will amplify this. Okay.
Okay, sir. Okay, sir.
The
Actually, based
amplification
on
will go on both sides, sir.
yeah, both sides. Yeah,
Sir,
both sides they will.
what if this fragment MSc1 and MSc1 fragment gets adapted by two MSc1 FLP adapters? Then won't it be amplified?
Can you repeat your question?
Sir, if the both ends of this MSc1 fragment is bind by this MSc1 FLP adapter, then
That is not going to amplify actually. Only
Sir,
this one is going to amplify. Okay.
sir,
Yeah.
sir, we will use two primers which are exactly complementary to the in base pair length it is complementary to MSc1 and PhD2, sir.
Yeah, this
But
adapter is complementary. Adapter will go, first we will design adapter such a way that it will go to this restriction enzyme. It will go and bind these sites. Then after that we will design primer for this, having
base
selection,
pair
actually
length same.
selection nucleotides. Then they will amplify this. Understood now? First, see here I think, first after your restriction fragments will generated the like fragments. Then restriction fragment then ligate it with an adapter molecule that contains the specific sequence that allow PCR amplification. Then adapter ligated DNA fragments are then subjected to pre-selective PCR using primers that targets the adapter sequence. Then the PCR chip enriches the fragments that are adapter ligated to both the ends. Then pre-selected fragments are then subjected to selective PCR using the at 3' end with 3 selection nucleotides. Then they do gel electrophoresis, then visualization scoring. It's just simple. First, treat with restriction enzyme. Those sites having both the sites of the different restriction enzyme, we will ligate the adapter, then use this adapter as a complementary sequence for the primer. Then we will use, then we will amplify these fragments. Then we will look at the polymorphism. This is called as AFLP. Any doubt? The doubts cleared or not? Either just once read this. If you not understood my explanation, just to once you go through this slide, I'll erase all things. Any doubts?
Sir, I'm getting confused that how will it identify that those adapters two are same or not?
See, we designing now. The adapters are specific for this different restriction sites. How they will get confused? This adapter is, see, MSc1 specific adapter will join here only. The PhD1 join will here only. They having complementary sequence now.
Not that one, sir. This one, see,
No, sir.
this
Sir, he is
one,
asking that
PST.
how can we know that there are two restriction enzymes? It can
Sir,
cut. How can we detect it?
but in book they have given radioactivity or fluorescence. With help of them, we will detect, sir. By
Actually, we have to
lifting.
use that actually. Then no, actually no, we are using two different. Before that we are not going to know. We will add. We already know which restriction enzymes are we are using. Okay. Then you just give the adapter. You just add another adapter in a PCR. It will happen in PCR only. See first you should have a append tube. You will add first DNA, then restriction enzymes. Okay, then it will cut. Then you will add adapters. You'll add all adapters like this one, PST1 adapters, MSC1 adapter, and then MSC1 adapter, MSC1 adapter. Here they will go bind, and PST1 adapter, PST1 it will go bind here. Those having this having here here MSC will adapter will go and bind, and here PST1 adapter will go and bind. Then after that, you are amplifying. For that adapter, we have to design PCR primers, and they are all different. How this can be? Because one primer, the PST specific primer go bind here, and MSC specific primer go and bind here, and this will only get amplified. How can we expect it will go and amplify? It can easily detectable now because we are adding all, but wherever complemented, then only. Yeah, these are you are asking this
Sir,
if
sir, what?
MSC1 adapter and MSC1 adapter, this cannot be identified or not. Actually, it is radio labeled. Okay, sorry, I missed the point. Radio labeled.
Sir,
We are
one more
having the
doubt.
different colors actually, so that we will detect them. Those having red and blue, here red, blue. Okay, I missed the point. Based on radioactivity, we'll use radioactive this one. Okay.
Sir,
The adapter
another
having
point.
yeah.
The primer also has the adapter sequence plus the selected nucleotides, sir. We will increase 1, 2, 3, sir.
What?
The primer will also have the adapter sequence, sir, plus the selection nucleotides, sir.
Exactly. Within
Yes,
the primer,
sir.
there is a selection nucleotide.
Yes, sir.
Within the primer.
Yes, sir. That's my doubt, sir.
Yeah, within the primer only selection nucleotides. Now your
Yes,
doubts is cleared. Actually, ask the doubts. Sometimes I used to forget. Okay.
yes, sir.
We should add some radionucleotide adapters.
Sir,
Actually.
can you explain this selection nucleotide again?
The selection nucleotide is nothing but, for example, A, T, G, C will be there. One should be fixed, not the degenerate. One should fix as A only. If it is, this is one selection nucleotide. Another one you fix A, and another other sequence are random. Means these three selection. Means the sequence having these three nucleotide in a, like for example, T only get amplified. In this here, the sequence which having the first T only get amplified. Okay. This is the only the selection nucleotide. Means it will reduce. Means how many fragments you will go to get all 3T together? It's very less. How many fragments having all first having the T end? It's very less. Then again, okay, some are there here 100 fragments are amplified. But again, you are using 3 selection nucleotide. Having 3 T's together, the probability is very less. Probability is very less. Then means your fragments number is getting reduced if you use some selection nucleotide. Means the probability of together of these 3 selection nucleotide is very less in the whole genome. That's the logic actually behind this. Now we got the point.
Yes, sir.
Actually, markers are tricky actually. You should imagine all the things. Those who are working on gels, PCR, these things, easily they will get. But those who haven't seen the PCR, gel, all those things, It's very difficult to imagine where they will run, how it will look. Everything happens in a single Eppendorf tube. Okay, you just all these components, then allow them, then do PCR, then run the gel. Simple steps. Okay, no need to do this all the thing, like understand these things in lab. Okay. Then another thing is sequence tagged sites. It's a means here 12 to 15 base pairs of end sequence of cDNA genome. Use the sequence information to design. Actually, it's a very simple marker. I told about scar marker. In scar marker, from RAPD gel, you are eluting some band and you are designing the end primer, then using them as a primer. Then you are amplifying. And you know that this fragment is where it is located in the genome, we don't know. Where it is there in the genome, we don't know. Then you can call it as a scar marker or any cap marker. But if you know where this particular band is on the genome, for example, it is in the fifth base pair on chromosome number 1. Like this, if you know where the band is coming from, where it is located in the genome, if you know, means it's a tag. It is the position of this band is tagged on the genome. It is tagged on the genome. That's why it is called as the sequence of the particular band is tagged on the particular genome. Then same, we are designing end of this one, then using as a primer, then use as a marker. This is similar to this one, but only thing is we know where it is located on the genome. The only difference between the scar and the STS marker. Understood?
Yes, sir.
Then another marker is microsatellite. Don't worry about STS, scar, all those things. Nobody is using. IFLP also nobody is using. Don't worry. But only thing remember either it is dominant marker or non-dominant marker. What is the advantage of AFLP? I told like it overcomes the disadvantages of RAPD and RFLP and combines the advantages of both the marker system. Should remember this much. And procedure. First, what you will do, restriction engine. Then you will go with amplification with selection nucleotides. Then you will get some reduced representation of the bands. Then you can able to distinguish between the two individuals. This is the AFLP marker system. And STS means similar like scar marker, but we know where that marker is present on the genome. Means where by cloning at the time they used to clone that one where this particular sequence is there. They used to get to know. Okay? That is a STS marker, tagged marker. Then another most frequently and even everyone is working on this marker is SSR marker. Very, very important, simple sequence repeats. In the name means, I think many of you know what is repetitive DNA. I already discussed what is repetitive. We having two types, mini satellite and microsatellite. Can anyone tell what is mini satellite and what is microsatellite? You know this one only, you can able to understand what is SSR.
So
What is the difference between?
In many satellites, there would be base pairs repeating of 10 to 20 kb, sir. But
yeah, it's a very large. It's a large repetitions. It's a very minor repetitions. Like AT repeated 10 times. ATGG repeated 11 times, 50 times, 20 times like that. But here, some KBs of sequence repeated in millions base pairs. Like in several KBs, several thousands of KBs times, the particular DNA was repeated. The sequence was repeated in many satellites. Okay, it is mainly in like near centromeric region, telomeric region, you can find that type of sequence. But microsatellite distributed throughout the genome. Okay? And I already discussed like different like maize having how much repeated DNA, rice having how much different. You have to know all those things. Then this SSR markers, you can see that For example, this is parent A, parent B. In this parent, this particular sequence in a one band, it's repeated 5 times and 7 times. In B, it is repeated 4 times and 6 times. You can check the progeny. Here 4 and 5 is there, 4, 7 is there, 6, 5 is there, 6, 7 is there. You can look at here, individual one, the different band size you are getting. Because it's having 4 and 5, see the bands, see here 4 and 5. And here, now with this system, we can able to differentiate all the progenies or not, based on the number of repeats. Like for example, 80 here repeated 5 times in individual parent 1. 80 repeated 4 times in individual 2. In this progeny 1, 80 is repeated like 5 and 7 and 4 and 6. And in 80 it is 4 and 5. In progeny it is repeated 80, 4 and 7. And another progeny is repeated 80, 6 and 5. And progeny 4, 80 is repeated 6 and 7. See, by this, if you have the each and individual, there is a difference in this particular locus having a different repeats of this SSR, like 80, anything. Okay, these repeats are, the number of repeats is varying from the different individuals. Based on that, we can able to distinguish them. You can easily say, okay, this progeny, they are all different progenies. Okay. And you can also say that all of them having the bands of the parents. Means there's, you can say that all the progenies belongs to these parents and they are all different. Like this, you can able to distinguish. I think I have some other. Means they are the short DNA fragments, usually 100 to 200 base pairs, means the length is very short, and they're made up of tandem repeats of 2 to 9. Either AT is repeated 10 times, A to T, like ATGC 10 nucleotide repeated 10 times. Anything may happen. Means it's short tandem repeats. For example, this 280 repeated nearly 100 times. 9 repeated nearly 4 times. Here you can see example. GATA repeated 4 times and GA repeated 10 times. The same sequence repeated 4 number of times. Here same GA repeated 10 number of times. Means there are 2 SSR sites. Understood what is SSR? Not marker. At least you understood what is simple sequence repeats.
Yes, sir.
The basis of
Very-
polymorphism here will also be
Yeah,
length,
it's
sir.
basis in the difference in the length of the repeats is the basis of polymorphism of your SSR marker. If anyone asks you what is the basis of polymorphism, means the difference in the number of times a particular repeat is repeated. Particular SSR sequence is repeated. This is the genetic basis of polymorphism. And it's very easy to perform and it's very abundant because for example, maize having like nearly 30 to 40% of repetitive DNA like this, because most of the DNA is repetitive only in human genome. Like in not only any genome, the majority of the part is repetitive genome. Means SSRs are more abundant. Then it's a codominant marker. It can able to distinguish heterozygote identification from the dominant homozygote. Then, actually, these points are somewhat contradictory. Many people are got confused. In one book, it is given SSR can be used with only within the species, but in book it's given can also use for across the species. Don't worry about this point. Okay. Then variation in the number of repeats arises due to unequal crossing over and recombination slippage. I told these repeats are possible. Why this difference arises between these? Because sometimes if there is unequal recombination, Here, normally should have equal proportion change should be there. But instead of 4 and 4, you should give 4 and you should give back 4. You see, 4 repeats. But if there is unequal recombination, here 2 only remains, but it will having 6. It will get 6 repeats, but another progeny will get only 2 repeats. Because of this unequal recombination and replication slippage, many of you know what is replication slippage. By that sequence, we will generate SSR getting raised, and we can use that. We can exploit them as a marker system, and it's very amenable for multiplexing. As I told, what is multiplex PCR? You can design by using you can run single two to three different marker at a single time if using radio labeled primes. That is can be amenable for SSR marker. But what is the disadvantages? It's very costly to develop. We need sequence information. How will you get to know? Here AT is repeated, GATA repeated 4 times, GA repeated 10 times. How will you get to know? If you have a sequence, then only you will get to know. Okay. Then another marker system is ISSR. Means I told about SSR. Means what are the sequence here? For example, you think SSR1 and SSR2. The intermediate region between two SSR marker, it is called as ISSR marker. Okay? For example, here AT is repeated 10 times, and here it is repeated, AT repeated so many times, 10 times. Then what is the region between the two SSR marker? That is called as ISSR marker. Then for designing SSR marker, we need a prime. We need a sequence information. For designing ISSR marker, we no need of sequence information. Why it is so? Because you are randomly designing primer. For example, you think that I will think GA. If you give 5 times, you can easily design by yourself. GA, 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 and GA. You design primer by yourself. You add to DNA, like in a Pappendraft tube, you do PCR. Means somewhere it will going to bind in such a way that if you are lucky, this sequence, this particular DNA fragment, in this end and this end, having this same sequence. GA, 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 GA. Here also you having the same sequence, and here also you having the same sequence, and it will go bind here, and it will go bind here, and the region in between them are amplified. Means this ISSR, no need of any sequence information, and it can bind to anywhere. It is not like that. This is here only it can bind. Okay, wherever it finds its complementary sequence like this, it will go and bind. And this possibility across the genome, n number. Okay, this type of sequence, this type of chances, maybe you get 10 chances. Hence 10 bands you are going to get. But from which locus you are getting that band, you will go no don't know. Okay, understood what is ISSR? And it's a dominant marker. It also unable to distinguish heterozygote and homozygote. Understood what is ISSR?
Yes, sir.
Means you just remember what is it's a multi-locus and it's a dominant marker system. It's mainly used to identification of genetic diversity percentage. These are all similar to every marker. Okay? Then it is located between the adjacent and opposite oriented microsatellites. Okay? Between the two SSR marker. Then what are the region you are going to get? This is ISSR. You randomly design anything. Okay, GA 10 times and here GA 10 times. Somewhere this possibility will be there in the gene. If you get possibility, you are going to get this particular intermediate region will going to get amplified. This is the only thing in the ISSR. What is the advantages? Means we don't need of sequence starter because you can randomly design the primer. Then low quantity DNA. If you have, if you need sequence specific complementary, we need a high quality DNA. But you don't know the sequence wherever it's going to random. Then if you low quantity DNA also you can use. Then the random distribution throughout the gene. Because the many times at least eukaryotes more than 40% of the genome is repetitive. Somewhere you are going to get this type of uh, repeats. Somewhere its possibility is very it's there. You can't neglect. Okay, no, it's no possibility of getting GA10 and oppositely GA10 and no intermediate sequence is not there. It's not. We can't say that it's not the possibility. It may be possibility is there getting the sequence like this. Then it's amenable for automation because it's a PCR based marker. PCR is automated. Then it's as a multi-locus. We can't say that they are the two different bands are there due to allelic difference. Because maybe different locus may be getting this different bands. Okay. We can't say that there only difference in the alleles. That's why we're getting two different bands. It's not like that. Maybe there is a change in the locus or a change in the sequence. We are going to get two different bands. You can't generate allelic relationship here. Then it's a dominant marker system. You can't use it for identification of F1 or the heterozygous. And maybe here the concept of homoplasy also arises, same as like RFPD. Means the similar sized fragments may not be homologous. Maybe fragments are getting similar size. You can think, okay, these are from the same locus. It's not possible. Maybe because of this having same size, you can't say that they are from the same locus. Means this homology fragments will always disturb your results. It's called as homoplasy condition. It's one of the biggest problem in RAPD and ISSR also. Understood what is this disadvantage and advantages of ISSR marker?
Yes, sir.
Actually, I am trying to at least somewhat like make easier these things because these are very difficult things. At least I am trying my level best to make you understand. Then I think you have an idea what is PCR and PCR based markers. Let me show some of the images regarding markers. See, this is actually my SSR marker, only what I have run. Here. My screen, you are seeing my image.
Yes, sir.
Now you are seeing what I am doing, editing. You don't. See, this is you are seeing my presentation.
Yes, sir.
Here you are seeing SSR marker. Actually, this is SSR marker. These are the two parents. Here parent one and this is a ladder. See, we are having SSR marker 1. Like it is a RM 493 something you just RM It's in RISE RM490. Like you are running in parent 1 and parent 2, and you are getting this difference. You are able. This is a polymorphic actually. First, you will do polymorphic survey. You can do this marker is having some difference. Now you can able to see the difference what I'm showing.
Yes.
And how you will get to know the best pairs means you are running at the same time you are running on ladder. Here in ladder we know where this here 100 here like here we are getting 50. Like this ladder position we know where the band is there and their position like what is the bands like weight we are knowing already. If you're getting position here to that, like you can say this is 70 base pair and this is 60 base pair. Like that, we can able to identify the base pair sequence, like what is the weight of that particular molecule, we're going to know. You can able to see the difference between parent 1 and parent 2. Then based on that is the progenies, progeny 1, progeny 2, progeny 3, like this. Like this, we are able to distinguish between the progenies. See, here it is like heterozygous. This is like parent 2. This is heterozygous. This is heterozygous. This is parent 2. This is, I think, parent 1. This is heterozygous. This is, I think, parent 1. This is heterozygous. This is heterozygous. And this is parent 1. This is heterozygous. Heterozygous. Parent 1. Heterozygous. Parent 1. Like this, we are scoring. This is the only use nowadays. Everyone is using for this purpose only. Now understood what is marker, what is how to do this. I hope you got. Yeah.
Yes, sir.
I think now we are clear. What is RFLP? What is RAPD? And what is SCAR? I will discuss while after discussing SNM marker. SCAR and CAPS. CAPS we will discuss. I think you know what is SCAR, what is STS, what is SSR, what is ISSR, what is AFLP, what is another marker we discussed. DNA amplification fingerprinting. I think we have covered this many number of markers. Okay, these are the only important. There are n number of markers which are there in the A guessing and B guessing. No need to read also. Okay. Another marker you have to read DART. I will explain now. Okay. Any doubts in these marker systems?
Sir, SSR marker is not multi-allelic. Sorry, multi-locus.
No. It is locus specific. It is multi-allelic and locus specific.
Okay, sir.
Because we are knowing the sequence now, like we are knowing exact sequence of that one, and we are designing based on the sequence information. That's why it's a locus specific and multi-allelic. Maybe SSR may having the many different alleles, but all alleles map at single locus only. Okay.
So we will design the primer that exactly correlates to a single locus.
Yeah, exactly.
Yes,
I think I will
sir,
show one website. I think Gramine, you will get to know how we can find the markers. Wait, I'm sharing on this one. Actually, this is the one. I think you are getting one website, Gramine. Everybody can able to see. You're able to see or not? This website.
yes, sir.
Yeah. Actually, this is one SSR tool where we can design the primer, can order the primer. Okay. For example, like it's a Gramine, it's having all Gramine species like Graminaceae species. Then, for example, marker home. Like this, we can have SSR marker sources panel. Like we have so many tables like this. For example, if you know any markers, SSR marker resource, you just type it. Then there is one Excel sheet will be there. Tab limited. See, like this, locus we having. See, this is locus, and where this is primer name, like for primer name, forward primer, and reverse primer. And I will show a supplementary table I have. There is a one table will come here in this only. Every crop we have database like this, at least for those who are having very good database like maize, rice. Here only it's having. No? We'll having one supplementary table. I think if you know this table, you'll get to know. You know, to do one markers workshop, people will charge 12,000, 15,000. Waste actually. Don't join any workshop like this. Waste of time. See, for example, this is will be the sort ID, like marker ID will be there, chromosome will be there. Like which chromosome actually something then distance next marker, which on which chromosome is present, how many number of copy number it will going
Not
to
appearing,
having.
sir.
Sir,
Hmm?
table is not visible, sir. The
Okay,
site
okay,
is
I will. Okay, okay, I will do on. Now,
Yes, sir.
now, yeah, you can see that this is the marker. Now names will be available. You can see how many number of markers will be there. See, nearly 18,000 markers are there here. 18,000, 830 markers are there. You can see the sequence. Like for this marker, the repeat is AG80. And how many number of repeats? It is 15 repeats. Which chromosome it is present? And this is actually, I don't know. This is one ID. Then forward primer, reverse primer. What is the product size you are going to get? Like after doing PCR, what is the size of PCR fragment you are going to get? Then what is the position? SSR start. This is on chromosome number 1, 2000-2315 to 2000-2374. This position. Okay. We know all this information. Okay. This is how the SSR markers is. Okay.
So that
Now I think you
position
have an
is
idea.
base pair, sir.
Hello?
That position you are saying is base
Yeah,
pair,
it is
sir.
in base pair. It is in base pairs. This base pair to this base pair. This AG80 has repeated 50 number of times in chromosome number 1. In another chromosome number 1, in chromosome number 1 only, in 2000, like 680-95 base pair to 2697 base pair, this 80 is repeated 40 times. Like this, we will generate this information when we having the proper sequence information. But this database may not available for all the crops. Okay, so orphan crops still now this type of database is not generated. Okay, so for rice, those who are working rice, I think many of you know this one. Here only we are ordering primers. There are so many things we can do. For example, from this list also we can design the primers. Like we'll order, okay, you select the primers based on your needed positions. For example, those who are working on genes, okay, your gene is present on chromosome number 1, this position to this position or near to this position. You order these primers and you do your evaluation in lab. This is the only thing we are doing. It's not like any big job. Okay.
sir.
Now,
One doubt, sir.
yeah.
Sir, we will anyways outsource the primers, sir, isn't it, sir? We will get the primers from companies as well. We will outsource it only now, sir.
Yeah,
They
we'll
only
outsource.
design.
Yeah, it is
Sir,
outsourced.
that is not under our study, sir, but I am actually curious to know whether they synthesis DNA, sir, according to the primer
Yeah,
base
they
pairs.
synthesis. You know, the cost is per nucleotide base. They will synthesize.
Okay,
Synthesize the
sir.
nucleotide.
Okay.
You will get like powder like mixture. Then you have to dilute them. Then you have to use. Okay. You like small powder like thing. The primer in
Okay.
powder. Actually, powders will come. You have to add water. Then you have to dilute. Then you have to create stock solution. Like many things were there. Those who are working
Okay.
already, they are getting.
Right.
Okay.
Hello, sir.
Yeah.
Sir, in DNA sequencing, sir, I had a question. Means when we go for sequencing.
I will cover sequencing in the next coming classes. If say start saying something, everyone get
Okay,
confused.
sir.
Tell
Okay,
your
sir.
doubt. Okay, I will at least came to know what is your doubt.
Sir, in DNA sequencing, we generally go for any tissue crushing, leaf tissue crushing. And
Yeah,
sir,
we are going.
so yeah, then sir, it is not like that key only one cell is coming and only one DNA is coming. So when we are going for any kind of NGS
That
or
we will.
third
Yeah, that
third
we will
generation,
discuss.
so how we it is ensure that only we are not over representation of the bases and only one DNA is sequence, and how to remove
See,
all
you are
those.
wherever the complementary is there, that only you are getting. Na, I will discuss what is depth. This is how many number of times your base will going to sequenced. What is the terminologies? How you do sequencing? If you know the procedure, we'll get to know these things. It's not like that. See, wherever how you do sequencing, can you able to tell the steps? So the DNA is same in all the cells. There is no change in the your DNA from any cells, DNA from this cell, DNA from flower cell. You take any cell, DNA is the same. Okay, the size is the same. You are doing PCR, you are getting amplified. Then only it's not like that. DNA when you have a different DNA in a different cell, that should be the worry. The DNA is same in all the tissues. Why you are worrying? Either DNA coming from this cell or this cell or this cell.
Okay, sir.
And you are fragmenting the DNA. Okay, you are isolating the DNA. Then maybe your doubt is the full for example, that 430 MB of DNA from one cell, again 430 MB of DNA from another cell. It will give 830 MB. Then how will say okay DNA from one cell is sequence? That is your doubt, I think.
Yes, sir.
No,
Yes,
no,
sir. Yes, that is it.
no. Actually, if you're doing RNA expression, now they have techniques single cell RNA expression, single cell transcriptomics, single cell genomics. Because RNA is different at different cell, we are making at single cellular level, we are isolating RNA from that single cell we are doing. That is another thing. But DNA is same. Okay, you are crushing all the DNA, we are joining the pieces. Like while joining the pieces, like if you know what is Kamer graph, all those things, then you will get to know what is that one. I will not go in detail about that. You read, there are so many videos are available. You just go through it. Your doubt will get cleared. Okay? It's a good doubt actually, but if your DNA is different from all the cell, you should worry about that. The DNA is same from all this, even though it is mixing. What is the problem? Because we are fragmenting and we are in adapters, we are doing PCR, we are making Palani PCR, then we will sequencing. If you watch Illumina website, like Illumina YouTube channel, I think so many sequencing, how they do Illumina sequencing in detail with animation, it is there. Now we'll get how we'll get at that time we'll get to know how to okay. Okay, bye. This much I want to convey. Tomorrow maybe I will discuss dark technology and SNP markers. Then I think tomorrow and day after tomorrow we will wind up marker class and I will start mapping population. Mapping population. I think one day I will cover. Then I will start QTL mapping and all this. Okay. Any doubts up to here? Shall I close? Shall I end the class?
Yes, sir.
Sir,